the discussion related to the concepts to this topic of the proteogenomics. So moving on, so what is this proteogenomics approach? So as I mentioned that it is genomics as well as proteomics. So before that, I think every one of you knows what is genomics, right? So genomics, so first of all, let us break this whole words into it. So genome means you know that the genes present in that, and ome means that the genome, proteome, transcriptome means all the genes, all the proteins, all the transcripts, all the metabolites. These constitutes the terms like genome, proteome, transcriptome, metabolome, and omics is the study of all these things. So now this the time has come when we are going into this era in this of the proteogenomics. So single omics. So you all know that the starting of the human genome map led to the field of the genomics, but this alone genomics is not able to answer all the questions, and that's why still so many diseases, be it cancer or any infectious diseases, all this remains unanswered. And so that's why now we have started, people have started combining the two powerful approaches. So what is the proteogenomics approach? So here in this simple diagram, I would like everyone to understand the concept behind it. So what is this? So for example, there is a tumor sample. Now how you will do this proteogenomics? So from tumor sample, you would extract out the DNA, which will be your whole genome sequence, as well as you can perform the whole exome sequence, and the RNA for RNA sequencing, and the protein from the same sample. Now, how this two needs to be? Just wait one second. I think there are some people who would be joining, waiting to join. Yeah, hopefully I've allowed everyone. Yeah. So now the concept where it has been integrated or these things. So for example, in the proteomics. So now let us go into this. Uh, Yeah, so for the proteomics, what we need is that if I think if you are some basic idea related to proteomics, so when we or if you so when we generate the protein sequences, or if you have attended the map session, I think you all have got some idea related to what is proteomics. So proteomics, when the peptides, so the in the bottom of proteomics methods, when the peptides are run in the mass spec, the ion peptide intensities or the signals in the form of the mass spec, the ion intensities, when the signals are we get the theoretical spectra. So we try to match with the experimental spectra. So now I think every one of those, if you have attended, so you will know that the theoretical spectra in the proteomics is what we get from the database of the human proteins. But that human protein that is present or from the databases like UniProt, that does not correspond to every individual's or does not capture the every individual's genomic signatures that are there, and which corresponds to the proteomic profile or the protein signatures in a particular human being. So for example, whose DNA, whose tumor have been who has got the disease, so that person's DNA signatures of the genomic profile would be much more different and will not be captured by the unique reference human proteome database that is there. So that's why the concept came of this proteogenomics, where you will take out the DNA whole genome sequence or whole exome sequence, perform the whole exome sequence, forget the somatic mutations, the copy number variations, the microsatellite instability status, etc. RNA, you will get the gene expressions, splice variants. Then from with these, you will make a customized database So that database will be at an individual level. So now you can understand the resolution that you are doing from the whole common reference database. You are now putting down the resolution to a single person with a single personalized database, which contains all the information related to that particular patient's genomic signatures. And then the proteins you have got or identified, the peptide mass spectra now will match with that person's personalized genomic from the database that has been created from that person's genetic profile. With this, you will be now scoring and then validating, and then you will get the sample specific peptide sequences which can be mutated on the basis of that person. So all these things you can capture in a simple by integrating this whole method, which can lead to you for the identifications of different novel biomarkers, different novel immunotherapeutic targets, novel pathways. For example, the peptides, the mutated peptides, if you can target that peptide to be an antigen and you can generate antibody against those, for example, the concept of new antigens, then this proteogenomics approach can help you to play such a role. So this simple block diagram, what is saying about the proteogenomics approach, but it requires a lot of both in the data generation as well as the analysis, and then you can get the overview whole picture in a patient specific or on a personalized basis. Okay, so this is what the proteogenomics approach is. Now moving on, so now this is one of the uh, revolutionary, means this a very good review article on the methods, tools and current perspectives in the proteogenomics. So now this gives a much more in depth that how this is done, that DNA, RNA after the sequencing, how you can go to that generation of the customized sequence database. But this requires a lot of steps and a lot of tools, which we will discuss as we go on. So for example, here you will need to have a, whenever you do RNA sequence, or you need to construct the transcriptome, or for the DNA, you need to alignment to a reference genome. The reference genome, now here is also a trick. The reference genome can be done no normal global genomes, that is the HG19 or HG38, which is present in which is present, but also it can be your uh, that from the peritumoral tissue or from the PBMCs. If you get the normal DNA, then you will get in a much more personalized database, much more personalized way, so that you will overcome the germline variants. You will understand the germline variants pattern as well. So generally, we are interested in the somatic variants or the mutations that arise in the due to the somatic, which causes, but you can get a pattern of the germline variants as well if you take the normal DNA also from the same patient only. Then you will do the variant calling, which include to identify the SNVs, indels, gene fusions, splice junctions, RNA editing, etc. And then you will extract this peptide spectra and you will do the same. So now finally you can either map your peptides to the genome, map genetic variants to proteome, do a map peptides to species. So this proteogenomics approach is not only applicable to for human species or for diseases like cancer, but it can also be taken out like the metagenomics for identification of the different microbes in a soil or in a community, that what is there. So you can do a metaproteomics kind of study where you can map the peptides to species as well, and how the effect of one organism present in a soil can affect the its influence other microbes in that particular community, or the different mutations that are happening. So and also you can understand at the peptide level how the genetic changes are occurring at the protein level, which is generally at the highest level and the phenotypic level. So now then there are different, you can perform the clustering, predictive modeling, pathway and network modeling. So all these can be done after the initial proteogenomics integration, and you can do all sort of clustering that samples. You know that in the cancer, generally World Health Organization takes a huge role in deciphering the clusters, or the what are the numbers of what are the numbers of uh, uh, grades, but it generally doesn't define in an histopathologically, and there are different debates. But that thing you can do in a clustering, co-clustering, multi-omics clustering. Also, you can identify the diagnosis, prognosis, drug response, toxicity, etc., etc. So then in the enrichment and all these things can be done. So now we will focus mostly on the proteogenomics only. These topics are quite different, and it can be done in a different series if you all want. So this uh, yeah. So I hope that there is no question till now. If there, please feel free to ask. So I'm repeating again. So feel free to discuss or comment or anything. Yeah, so now another thing is that one can understand that this is a very unique thing, a very important thing to understand the mRNA protein correlation. So Achha, yeah, yeah, any question? Yeah, yeah. So if you Yeah, so if you try to work on a, for example, so actually it's the genomics data, the difference. For example, if you do a proteomics study and take the reference database that is generally taken for searching in any primary analysis, so you will get then overall the peptide proteins and the peptide, identify the peptides and the proteins. But if you want to know the cell specific, for example, if there is a heterogeneous group of cells and you want to map those things with a cell type that is actually which cell type it is co-clustering, or you want to map it in a very personalized way, then it is necessary to have both genomics and the proteomics data. Then you will get that particular specific specific peptides and the specific mutations. So for example, if you have knock down certain genes or something, then what is the effect at the peptide level, you will get in a better resolution, better resolved way. Yeah, so the so the genes, so that yeah. So what happens is that when you get the genomics data, when there is a, you will, how will you create the database? So you will perform a 3 frame or the 6 frame translation of that particular genomics. So whatever mutations or whatever they are in the sequence, so it will correspond to that in that way, and then in a translationally or in silico way, the peptides will be formed. For example, if you use the tryptic peptide, so it will form the tryptic peptides from those sequences that you have used for uh, using the with the mutations. So all those peptides that are mutated will be captured, and if that particular peptide is available in your sample, then you would be able to identify it easily. Otherwise, with the normal reference database, that peptide won't get identified, and it can be used as, means it can come as a contaminant or something else, but you cannot capture it because the peptide spectra, theoretical spectra that you are generating, won't contain that mutated peptide if you don't use the genomic data from the same sample. Yeah. Yeah. So it is clear, right? Yeah. So now the is the mRNA protein correlation is proteogenomics. So in it has been seen in various studies that we get in the whatever we get in the mRNA level, all those changes are not reflected at the protein level. So there sometimes there can be negative correlation as well. So this study has, for example, shown that the mRNA transcript level there has been increase in the expression, but in the proteome expression it is not so. So these kinds of relation or correlation or this complex type of things can be done or can be studied using the proteogenomics approach. Secondly is the copy number expression to the copy number analysis to the expression data that of the copy number variations is happening for a particular. So what is the effect at the proteome level? So this gives us a very good view of all the variations that the genomics level, whatever happens. So all those markers that like the gene mutations and all, like for the epigenetic changes or the genomic changes like the MGMT promoter changes for glioblastoma or NFT mutation for meningioma, etc., etc. But what are those effects at the particular proteome level or the protein level via RNA level? So all the things in the central dogma is captured very much well using the combination of these approaches or quantitative trait loci to miRNA regulation. All those transcripts and miRNA has a very important role in the regulation. So there are various multiple steps, and only by looking at the DNA level, we cannot say that the mutations then signals also the mutations of different types. So we will discuss it in a later part. So everything can be captured using this, and it is an emerging technique which all over the world people are trying to for resolving the complex diseases. So this is an example of the proteogenomic characterization of the human colon and rectal cancer, which was done uh, by the group of the TCG and the CPTEC. So we will look at the we will look come up to all those things. So this is like the TCGA is the first consortium to that started using the cancer genome atlas, followed by then Cosmic is that which catalogs all those mutations in cancer, which is found in the TCGA using the normal reference genome like AG19 or AG38 now is the normal genome. Then the DBSNP, which is your database of the single nucleotide polyform. So here actually people are mostly interested to look at the single amino acid variants or single nucleotide variants at how it is changing, because that single amino acid change can also change the nature of the protein. As we all know that the protein is consists of amino acids, and amino acids are of different types and branches. So one acidic amino acid changing to basic amino acid can have a deleterious or detrimental effect on the protein overall. So these, and also in the cases of drug discovery, if you target a particular protein but its structure is different because of the change in the mutations in the cancer, so your drug becomes ineffective if you don't know the actual amino acid level changes and the structure of the protein. That's why also sometimes it happens that the drug doesn't uh, in that protein in that particular patient. So all these things can be captured in a very good way in a proteogenomic and a personalized way. Second is, so these are the some of the results from those which they have performed. Uh, yeah, yeah. So these are some of the results that they have showed. But so this just put them to show that how different things can be captured with these types of things and with the databases, and these are with the commonalities they have found with the normal and method. So for example, so impact of the SNVs on protein sequences is huge. So I'd like to just highlight that all the yeah, so all the non-synonymous protein coding SNVs detected from the were annotated. So their existence in DB, SNP, Cosmic, and previously reported. If the SNV is included in with sufficient spectral count for quantitative, we quantify they quantified the impact of the SNV and protein expression by level of the genes in the SNV containing sample, and then they given us code how they found out the impact of the SNVs. Another thing is that the correlation between the steady state mRNA and the protein abundance. So how when all mRNA and protein measurements, then they use the FPKM and the SNSF value state estimate your mRNA and the protein abundance. So the mRNA transcript level, the abundance in a particular individual particular sample, how much is the expression level in the protein, if it is correlating or positive, or the no, negative correlation is in the expression level. And the second is the protein mRNA and the protein variation, that how the genes with minimals, how much they calculate, they got the difference in the variation in the proteins when it was done. So this protein abundance and variation, so both in the expression of the fold change and the variety in the structure and the nature of the proteins, one can understand. So this is the proteogenomics approach. Now we will go to today this session. We will try to look at the individual things of the genomics, especially that how the sequencing technologies, what are the different platforms that are being used now in the current state, what are the platforms that are used, what are the principles behind the sequencing technologies, how the improvement of the sequencing technologies have helped to gain momentum in this field, and how the computational tools are also playing a huge role. So the field of, if you know, the field of bioinformatics has emerged from the late 1980s with uh, different databases coming up with the NCBI, PDB, and all. But now, how 1 second, so another request for, yeah. Yeah, so for the genomics, if we look into it, so it is the sequencing technologies that has played a huge role and the part. Then it is the analyzing the data, and then is the interpretation of the data. So sequencing methods. So what are the sequencing methods if we look from the history till the current version? So first was the Sanger sequencing method, as we know. So this is nothing but a chain termination method, which generally takes your original DNA and then it gives the DNA. So here I understand the different sequencing technologies that previously used, though, and was very successful and very uh, popular to, was a Sanger sequencing, but though it is not being used currently in this high throughput technologies and the cutting edge technologies and the instrumentation that I've done. So we will go one by one to each. So firstly, there's a Sanger sequence. Let us still revive the principle of the Sanger sequencing. So the Sanger sequencing is that which uses a mixture of the DNPs and the but this helps us to understand that the how technologies has evolved with the principles of the chemistry, and how this thing is being taken in a cutting edge with the the cost being drastically reduced for sequencing, and giving us an idea how the whole genome and the whole exome sequences, and helping us to unveil all these characteristics of particular diseases. So now then when the fluorescently labeled oligonucleotides were there, and whenever it got inserted into it, it provided the small fragments, and the 5 prime to 3 prime this DNA polymerase could not synthesize it, and then it was size separation was done on the capillary gel electrophoresis, where you know that the basis of the molecular size. So wherever the DDNTPs has been inserted, so there you will get the size will get reduced, and in the from the gel you can finally, with the help of computational algorithms, you can detect the whole sequence. So this is a simple method which is also known as your chain termination method of the Sanger sequencing, or the chemical method of by which you can, with the help of DDNTPs, and you can understand the sequence of the genome. So now this is also called as your first generation sequencing methods. So next is your next generation sequencing methods, and this is what revolutionized the field of genomics, and whatever we high throughput coverage and the reads and all those things that we are able to achieve, has been done with the help of this methods. So you can check out that the Illumina, so you can go and search in their website as well, and you can find out various things. So I would like the discussion in the group to continue, that which has the which to be used for which application, that which machine to be used for which application. So I think that you all can search and look for and discuss, then you will get an overall idea that for which kind of what kind of application, for single cell sequencing application, like the single cell sequencing is now taking a huge place, rapid place, so which gives us the co-spatial understanding of the cells or the heterogeneities, and how their pathways, different pathways are affecting in a particular tumor tissue or the organ specific region. So we can understand. So these are some of the tool instruments like MISEC, HiSEC 2500, HiSEC 3000, HiSEC 4000, NextSec 550. So everyone has a different application. So I think the pros and the cons and the applications can be discussed in the group, and one can share and discuss and learn. So, but I'll go through the synthesis approach. So we should first understand this, how things are being sequenced. Then only we can understand the analysis and what are the reasons, or how we can get through it. We can understand in a better way. So first thing is that the step one for the next, so like in the Sanger sequencing method, that the DNTPs and the DDNTPs were mixed, the fluorescently labeled DDNTPs, and those gave the signal whenever there was a, okay, so just a minute, I'll let people in. Yeah. Yeah, so like the question. So I don't think is there any question till the Sanger sequencing method? So yeah, so feel free to, whenever you want to ask or comment and discuss. There's no issue. Yeah. So the next is here by the sequencing by the synthesis approach. So the thing is that like the Sanger sequencing, just mixture of the chemicals and then get the in the gel. So here it follows a different kinds of approach. So first step is what we known as the, yeah. So what first step is known as the, okay. So yeah. So if you have any question, you can raise your hand as well. In the so we will answer you. So so step one is the library preparation. So what is the library preparation? So here what we know is that since you are in the, just think of it in this way so that you get your concept clear. So that's why I told that the learning, what I feel or believe is that the learning in history helps us to understand and also helps us to understand that how the future technologies can be improved or the future technologies can be built up. So in that we used to provide in the Sanger sequencing, it was provided the ddNTPs, and with that one can get the fragments and also in fact, if you all know at the Human Genome Project, there was your the full your full your that fragments of the genome, and then it was the chromosome walking method which led you to the full sequencing of the genomes. On the basis of that, in the and the libraries were stored in the plasmids. So here you can just understand that how we will know that which fragment corresponds to where and how these things will be done. So that's why the using ultrasound or some ultrasonic fragmentation of the genomic DNA with 200 to 500 base pair in length is followed, and then the 5 prime and the 3 prime end adapter are ligated, so that the adapters, we know what are these adapters. So whenever these adapters, we know what are those adapters, and we are just ligating it. So in a process known as tagmentation, which is the fragmentation and the ligation occurring at a same step. So now I know the what are the adapter sequences. Now I can put them in our known adapter sequences, and from where the signals will get, we will able to match and make the whole complete sequence. So this is first. So that's why we need to have the adapter ligated at the 5 prime and the 3 prime end to know that which part of the genome is being sequenced and from where it is coming. So this is our library preparation. So first we will fragment this and we will uh, ligate this. Then is the step 2 is your cluster generation or pooling, where we need to amplify the genomic sequences. So you know that we need to amplify, so attaching the DNA sequences, so it randomly on the length of the source of the flow cell. So here flow cell is being used, matching with the adapter sequences are already there. Okay, so yeah, so I don't know this I have to figure it out that if it allows other person to let it in, otherwise it becomes sometimes difficult. Yeah, is a question? Yeah, I'll take. Yeah, huh. Huh. Yeah. Is there any question? Yeah. Flow cell. So flow cell is where your all those capture and the sequencing happens. Okay, so here if you the flow cell is that part of that where bridge amplification and means it's a kind of a what you say that semiconductor based thing where this all those DNAs are captured, and then finally after amplification you get the signals out of it. Okay. Just a minute. Hmm. Hmm. Yeah. Hello. Yeah. Say. Yeah, so in the library distribution, what is done is that the DNA fragments are first your fragmented, okay, in the range of 200 to 500 base pairs, so that the amplification can be done on a small strength of sequences for better sequencing. So after the fragmentation has been done, then the 5' to 3' end known adapter ends are ligated. So this way a library is prepared with which we know our known ends, okay, with which we know our known ends of the sequences. So then when we capture it in the flow cell, then it gets captured to that places only where the known adapter sequences gets attached, and then we can perform the PCR amplification or the clustering, and then finally we can sequence it. Okay, so that's why we fragment the fragmentation is performed, and so and the ligands are lighted. So this process is known as fragmentation, and this is how library is prepared. I'm I'm clear, right? Okay, so if there is no any questions, so I'll move forward. Yeah. So now the third step is the sequencing, where DNA polymerase, connector primers, and the fluorescently labeled DNA DNTPs are added to reaction. So if you have still doubts, you can ask in the question or the chat. Okay, so I hope that it is uh, clear and I'm able to make you understand. So there's the library and then the cluster generation, then the sequencing. So now the DNA pool will label DNTPs as added to the reaction, and then the whichever the because of the bridge PCR amplification and all the clusters, and the cluster means of the same sequences, you will get a bunch of the sequences which will be amplified, and then when the OH is basis is added for a particular that region, then you will get the fluorescence signal. So the primers helps us to track down that from where the sequence or what sequences we are trying to get the final sequences. Yes. So anyone has a question? Yeah. This is raising the hand, please. Yeah. Uh, Hadi, I think you have raised your hand. Please. Do you have any question? Okay. So yeah. So okay. So I'll continue. So you can ask. Okay. So you have asked that why both genomics and proteomics are mixed together. So I think I have answered this question. So this to capture that the changes in the genomic level of a particular individual, how they are being captured at the proteome level. So proteome level is the phenotypic expression of what genomic signature is there. So the whole by integrating these two things, we can capture it whole. Okay, I think I am able to answer. So now let's move to it. So none of after sequencing, and when you get the sequences, so what happens is that you do a demultiplexing. So this step is done because that in the flow cell and in the during the preparation there are maximum multiple samples are being taken, multiple samples are sequenced at a time, so that every sample is given, every data set is giving unique molecular barcodes. So this helps us to identify that from which this step, demultiplexing step, helps us to understand that the sequence comes from which particular dataset, that it belongs to which particular dataset, that can be further led to further identification of the downstream analysis. So these are the four steps in the sequencing, in the next generation sequencing methods. First is your library preparation, then the cluster generation of the pooling by bridge amplification and the PCR amplification of all those sequences, then the sequencing by the DNA polymerase and the reaction, and then is the demultiplexing, and then final is the alignment and the data analysis. Okay, so the demultiplexing helps us to understand that what are the unique molecular barcodes and ensuring your accurate sample identification, and then finally going for the alignment and data analysis. Hopefully it is clear. And I would request everyone to go to the Illumina web pages and uh, 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 try to identify those this all those instruments, next sec, high sec, and make a discussion in the group that which one is used for which application and how it can be useful. So after this, another method is for the ion torrent by Thermo Fisher Scientific. So they used a slightly different principle than what Illumina is done. So Illumina is also one of the uh, revolutionized the field to a large extent. So in the yeah. So if anyone has any question, the hands, please. No, no. So this sequence is the flow cell. Yeah, so this sequence happens closely by passing on to a reaction, but multiple data sets are being taken at a single time. No, that's why there are these steps to further check on the quality, like the scores and all, that how what is the, and also the coverage levels. So all these things needs to be checked at a later stage before further analysis. Yeah. Yeah, so I can see something in the chat. I have to answer all questions. Questions and the last one. Yeah, so the course material I think is enough. Yeah, the course material are enough for you to answer all the questions, and also so that it's equivalent or something like that. Yeah, another hand I can see of Cyan. Yeah, yeah. So in the demultiplexing, yeah, in the demultiplexing, what is happening is that since there are number of samples are taken uh, for particular analysis, particular sequencing. So the unique molecular barcode is being attached for every data set, so that one can identify that from which data set is belonging. So just to check with the molecular barcode, and to just then find out the from actual data set where the sequences has been gone. So this is just a demultiplexing for multiple samples when it is being analyzed. So these are the four steps before we go for the final alignment and the analysis. Yeah, correct. That which sequence it is. Yeah, Hadi, I guess. Huh? One sequence. আচ্ছা
এই সেটআপটা একটু ঠিক করি না হলে অনেকেই অ্যাডমিট হতে পারছে না, বল 2 মিনিট দাঁড়াবে।
Yeah, so I think that just if it would be good if you mention your this questions in the chat. Otherwise, there's because people are also getting admitted and all these things, so there is sometimes lag in the question. Wait for 2 minutes. Yeah.
সেটা কি গ্লিটার সিলি করতে হয় মুশকিল
কি কোশ্চেন আছে দাদা?
না না, একটা কাজ কর, তোর মোবাইলে কোথাও খোলা আছে যেখানে আমি অ্যাডমিট করতে পারবো লোকেদেরকে?
মোবাইল
তাহলে খুব ডিফিকাল্ট হচ্ছে, নাহলে তুই করতে পারছিস না, বুঝলি না? হ্যাঁ, লোকেরা বেরোচ্ছে ঢুকছে বেরোচ্ছে।
হ্যাঁ হ্যাঁ
কোর্স ম্যাটেরিয়াল আছে তো, যেটা এখান থেকে কোথায় আমি করে আসছি
people যারা একটা অ্যান্সার করি আমি অ্যান্সার ফাইনাল আছে বার করে so the barcode sequence is already present in the organism. So actually, generally, what this is generally that some kind of tandem repeat sequence or something, which are generally not thought of to be present in the organism. It is used like the for this. So it generally does not cause the error in the identification of the sample. So all these actually barcodes and those adapters are well known to us beforehand. So this doesn't create a problem. Okay, so I'll start. If you have any other question, you can mention it in the chat as well as discuss it in a later in any group. Okay. Yeah. So now moving on. So this is the another thing or another instrument of ion torrent of the Thermo Fisher Scientific. Also, I would say you all encourage you all to go in search for the different videos of Illumina. Then you can find out you can visualize it well in a video format. So I have given you the basic concepts behind it, which includes all this. But yes, definitely you can refer to their videos for better visualization and understanding. I hope then it would be much more clear. But I would say that this is the basic concept. The concept is nothing very much difficult. It's just you need to have the sequences amplified for better signals. Otherwise, your signal strength won't be so much. So that's why you are making the small libraries of the fragmented DNA, and then you are making the clusters, and then finally you are sequencing it in the and getting the signal. Yeah, and then you are identifying multiple time to do with the multiple samples followed by the demultiplexing, and then you are doing the alignment of the data analysis. Okay. Yeah. Yeah. Can you repeat the question? No, this is the genomic. So, so actually, it is your depending on the means the genomic fragments which you are taking. So you are making the library of that. So if you are sequencing at the cDNA level, so then you will prepare accordingly. Yeah. So just repeat it once more. Yeah. Hello. Yeah. Yeah. Just repeat it once more. Yeah. Yeah. The procedure quite often remains the same with some modifications. Yeah. Huh. Okay.
এটাতে আনমিউট করিস না, এইটা স্ক্রিন শেয়ার কর, এটা দিয়ে করো, ঠিক আছে?
Yeah, so now moving on. So what happens in the ion torrent is that which helps you to detect the this detects the hydrogen ion or the change in the protonation. Or what happens is that here that where in the previous one in the Illumina we are trying to find out the fluorescently labeled DNTP signal we were trying to uh, record. So now here we'll be recording the pH change. So here what happens is that when the DNA polymerase incorporates your nucleotides or causes your pH changes proportional to the incorporated bases, then pH changes. What happens is that it is detected as your electric signals, and finally you will get the revealing the DNA sequence. So it does not require the fluorescent levels. So there is a slight difference between the principle of how Illumina and ion torrent occurs. So I think you can see check the pros and cons of it, and this causes your pH changes happening. So the pH changes is captured by the changes in the hydrogen ion. Third thing which is of so is there any so yeah, you can ask a question. Related to any cancer specific, this is to the related to the techniques. This is the proteogenomics. I have already given an example of how you can map it to the metaproteomics or the proteomics related studies as well. Now for the Pacific biosciences or yeah. So in the Sanger method, we are doing the by termination. First of all, we are you are using the DD method. DD you are adding and you are terminating those, and then you are using a gel, and then you are trying to match those fragments, which doesn't happen in a very high throughput way. Where in the Illumina you are sequencing it by synthesis method, where you are uh, sequencing, performing the sequencing and changing the you are in the fluorescently labeled DNTPs, you are measuring the signal and recording it in electrical. No, no. Yeah. Yeah, so the Pacific biosciences method, what it happens is that it takes your the single molecule real-time sequencing or the sequencing by binding method. So what happens in the SMRT sequencing method involves the incorporation of the fluorescently labeled nucleotides in real time as DNA polymerase synthesizes the DNA strand, and the NCV method incorporates native nucleotides and eliminates the need to uh, cleave your fluorescent modification. So it just incorporates, keeps on incorporating the native nucleotides and eliminates the need to cleave your fluorescent modification. So this Pacific biosequences is also helps you in getting the sequences in a very high throughput and rapid way. So this is one thing. Another sequencing technologies that is coming up and emerging technology method is the Oxford nanopore sequencing technology. So it utilizes the nanopore sequencing where the single strand DNA or RNA molecules are passed through the nanopores, and this. So now in the next generation method they use the signal based ion detectors changes in the pH level, detection of the pH level changes. And now here you can see that Oxford nanopore technologies, what it helps is that it generates characteristic disruptions in electric current that are used to identify and decipher the base sequences uh, uh, in the real time. Okay, so this with the within the nanopore sequencing, whenever there is a single stranded RNA molecule pass, so whatever the sequence which corresponds to the particular, so it causes a disruption in the electrical current, and then which helps us to identify and decipher the base sequence that in real time. Okay. So this is for the nano sequencing. This is very emerging technologies. I request you to go through this paper of the Nature Biotechnology, where you can get all those principles and those applications of the nano sequencing technology. And then another method is the 10x genomic technology, which also employs the infect sequencing. This actually is very much good for the sequential level. So whatever the sequencing technologies are coming up and emerging up. So what it happens is that it employs your link read sequencing, partitioning individual DNA molecules into your tiny droplets, facilitating the sequencing at your single cell level. So this is another technology which is being 10x chromium level, so 10x chromium sequencing technology, so which helps you to understand this individual DNA molecules at a very much single cell. So this is also a kind of and it is very much popular, I would say, in your immunological aspect or in the immunological studies of this sequencing. So you can find out this application easily. So I request all of you to go back and look at these technologies, what are the pros, cons, and note down that for what application are those being used. But I just here wanted to give you the flavor and the concept behind that, how these technologies are evolving and uh, how they are using. Now, for example, the BGI Tech Solution, so Beijing Genomics Institute is now offering a large scale of using these instruments, the large scale applications to uh, for the genomic solutions and all those. So the thing is that for the proteogenomics approach to evolve and to succeed, it all depends on the technologies, how the robust is that. So for this, we focused here on the genomics technologies first and how it has evolved rapidly and how it has incorporated the sequencing to a very high throughput manner in a very low time at a very low cost, and how the multiplex samples can be done at the same time using the different concepts or the different principles of like changes in the ion OH, pH, then the changes in electric current, changes in your fluorescent regulators. All these things from the standard sequencing method, how all these things have been evolved. So this is of the different methods of the technologies that are present. Okay, so any question, anything? So nano sequencing generally does not come under next generation sequencing. It is generally considered as for the third generation sequencing for those. Yeah, so I saw that I have answered. Any other question? Hadi, I you have a for hand. Uh, question, anything? No, right? Okay, fine. So moving on. So now once we are done with the sequencing, now what is the application? Okay, so what is the application of it? So first kind of application is to identify SNPs. What is SNPs? SNPs is our single nucleotide polymorphisms, right? So I here I'll go from some basic things as well as some advanced things. But basic SMP is different is that it is that the any sequence that is present in the normal, a single nucleotide level change is the single nucleotide polymorphism. So now that all those, so now we all know that what is the effect it can have in the further downstream processes like in the genetic code. So in the genetic, if the DNA sequence is changed, so similarly it will get changed in the genetic code as well, and similarly it will get changed in the protein sequence as well. Now the interesting part lies in that the from single nucleotide polymorphisms to single amino acid variants. So now there were many questions that why these two things has to be integrated or if it can be studied for other things. So now just in the scope of integrating these two sequences, integrating these two fields can lead you to understand from SNPs to SAAVs and the effect of those SNPs on single amino acid variants and further in the phenotypic level at the proteins. So that's what I gave the example first, that how their protein abundance and the protein expression variations are measured on the basis of these proteogenomics approaches. Okay, so this is the SNPs what we need to capture or identify after we are sequence a disease person with a normal person on a disease DNA with a normal DNA. That we get the sequences. Now, what are the SNPs? At what places are those SNPs? At what chromosome level are those SNPs? So how it can be there? So all those translocation, inversion, deletion, duplication, etc. All those things can be captured. So now, if you go for the basics of SNPs, so what are those kinds of SNPs? One is your biallelic SNPs. What is biallelic SNPs? That the two alternative nucleotides present in a specific position in the genome. If it is present at a specific position in the genome, if the two alternative nucleotides are present, that is gives us the biallelic SNP. Like one individual may have a C nucleotide and another individual may have a T nucleotide in a particular loci. So the what happens is that the C is changed to T. That gives us the concept of biallelic. And another concept is that triallelic or multiallelic SNP. So there, with the pattern, with all these all the sequencing things and the sequencing genomes and the patterns, one can get an idea of the and with the different algorithms, you can find out the spots or the places of the allelic sites where it is coming for a particular disease. Okay, so now another concept is the multi-allelic SNP sites or multi-allelic SNP, where three alternative nucleotides occurring at a specific genomic position at a particular locus. So what happens is that one person can have an A, another can have a G, another can have a T. If this sequence pattern is being observed for a particular disease, then we can say that it is a multi-allelic SNP site or tri-allelic SNP sites. Or so this is another concept. So we can have biallelic SNP or triallelic SNP. And then is the indel SNP. So I have not given the definition. I think you all can understand if there is an insertion of any nucleotide at any level of a locus in a particular genome or the deletion of any particular nucleotide, it's called the insertion indel SNPs. Okay, so this is the types of the SNPs that are present. Okay, so hopefully there's no question or anything. Yeah, so now after we get the single nucleotide polymorphisms, now comes the concepts of the mutations. So this single nucleotide change, as I mentioned, that this single nucleotide change will lead to the substitution of amino acid for another in the protein sequence. So if the protein, the amino acid is changed in such a way that there is no change in the if the codon is changed to the particular SNP in such a way that there is no change in the amino acid sequence of that particular protein due to, as we know, and I believe that you all know that the genetic code is degenerating in nature, and as a result, that mutation does not cause any change in the amino acid level, and then it helps you to identify. This is called the missense mutation. Which doesn't cause any change is the silent mutation. But it happens, if it happens that your mutation gets altered, the protein sequence, and as a result, your protein structure and the function has changed, then it was called the missense mutation, that the SNPs has led to change in your protein structure and variation. Then is that if it introduces a premature stop codon in the DNA sequence, then it is called a nonsense mutation. That okay, so now due to the SNPs, what happens is that your one that codon comes that which suddenly introduces a stop codon where it was not to be meant to be introduced stop codon, but it introduces the stop codon and your protein becomes truncated and it could not translate it fully. So this is called your nonsense mutation. Another thing is that if it is present in the shift, causing a shift in the reading frame, that then it is called your friendship mutation. And other things like duplication, inversion, deletion, all these things you can easily understand. So this is the types of the mutation. So it depends what is your mutation. So there can be silent mutations. So there are various SNPs which we can decipher at the genomic level, but no change at the proteomic level. That's why only considering the mutations. This is another reason why considering any other mutations we cannot find out. So yeah, so there's a question that what is the synonymous mutation? So synonymous mutation is that where you get due to the changes in the amino acid, which is similar, which is same to that particular amino acid, then it is your synonymous mutation. So that synonymous mutation also does not affect any changes in your protein level, whereas the non-synonymous mutation has your missense mutation and the nonsense mutation. So the broad definition comes under synonymous and non-synonymous, followed by this. Okay, yeah. So I would put my answer. Yeah, so this is an example. Let me check. Yeah. Okay. So just checking if someone is waiting. Yeah. So this is data from this is an example from a real data. So you can see that the SNP is identified is 312894. So by doing the different tools and the analysis, which is yeah. So the question was to study the data of fungus, fungus salt mass would be as most studied percentage of fungus. I could not understand your question. I think if you can reframe it once, it will be better. Yeah, so if you want to ask the suitable method for sequencing, so there's not so anything related to suitable method, but for different applications. In what I meant is that for the like the single cell application or the whole genome or the whole exome or the throughput or the coverage or the targeted sequencing, targeted exome sequencing for in those kinds of applications. For any kind of application, you can use any of those methods, but it depends on everything on your starting from cost, time, throughput, everything. Yeah. So if you understand the variance by type, so it will be so. For example, in this real data, so or the lecture. So the thing is that we can perform the analysis and identify the SNPs with the steps there after the DNA alignment and the analysis. So this is the example from a real data which we analyze. So we can see that the SNPs is identified, then MNPs, insertion, deletion, mix. So there was no inversion, duplication, or the interval. So total variance by type we could identify, then the number of effects by impact. So what, how much impact this variance cause? So the high, low, moderate, and the modifier, and then the missense, nonsense, and the silent mutations. You can see that nonsense mutation was 307, which showed that okay, so there was a stop codon inserted due to this SNPs. There is a silent mutation that there is no change, is a 46%, but 52% are the missense mutation. So in the genomic level, we can understand about all the these mutations, but what these missense mutations and effect on the peptide level or the protein expression level or the mRNA binders level can be understood through the by integrating both the things. So this is the example of the variance by type and how we one can get after the analysis, one can identify those variance and it provides. So there is a tool called SRPF. And so I think if you all want, we can discuss it in detail in different series, but you all need to mention it in the group that you all want those things. Then only we will be able to uh, arrange for those. Yeah, so this is for the number of the variants by type and then the effects by the functional class. So name next is the variance databases. So as I mentioned that in the paper which I showed as an example that they were understanding that what is the common and the unique variance that they have got for their particular analysis. So for example, there is one catalog of somatic mutations in cancer database, DBSNP database of somatic nucleotide polymorphism, which catalogs uh, all those which catalogs all those single nucleotide polymorphisms. Then there's a Go exome sequencing project to discover the novel genes and mechanisms contributing to heart, lung, and other blood disorders. Then there is an OpenSNP which allows the customers to perform genetic tests and publish the results. So you all can go through all these websites and find out the different catalogs of those somatic mutations. So as I mentioned that we generally people are more interested in understanding the genomic somatic mutations and not the germline variants. Generally, germline variants are not so much detrimental, but one with the whole genome sequencing and all, one can understand the germline variants as well. So the then the thousand genomes project and all those things have helped us to catalog a huge number of diseases, but still it cannot be reflected at an individual personalized level, which can be done only by only with the help of enough. Yeah, anyone else? Yeah, that's why I said that naturally occur, mostly occur in a population due to the germline variant. So that's why we are interested in understanding the somatic variants. That what has happened, not in a germline manner, but in a somatic mutations. So that's why we are want to analyze those, and then also the effect of those particular proteins, and that's why we need to identify in that disease context that how they are relevant, that in the particular pathway how those protein changes has led to the downstream cascades difference that has led to the or helped with of the disease. Yeah, hopefully I could answer it. Yeah, so now there's some, let us take out some understand some basic concepts of the units when we are set as I showed in that paper. I think you have all have been curious that what is FPKM, what is RPKM, and what are the meaning of this. So FPKM is that what is the, and also if, at the protein level and the mRNA transcript level. So see when we are going genomic is what kind of a qualitative where you can find out the mutations or SNPs and how many mutations are there, what are single nucleotide polymorphisms. But whenever you are going to the transcript level and in the proteome level, you want to find out the abundance level. So what is the abundance changes or the expression changes at the transcript level? So that needs to be have a quantifiable unit or quantification needs to be done. So here you can find out that unit which is through the mRNA transcript abundance is measured is your FPKM, which is known as the fragments per kilobase of transcript per million mapped reads. So this all the FP all the if you go to the full form of it, you will understand the meaning of this, that how this unit is calculated, FPKM is calculated. So you have if I will just there's a yeah, the fragments per kilobase of transcript per million map reads. So this is done to normalize. So FPKM and RPKM, these are all normalized expression data, normalized expression unit, which takes in account the both your transcript length and the total number of mapped reads in that sample. So that it helps you to that there is no discrepancy between the reads or the expression level when you are saying, so that it is normalized. So it represents your number of fragments, RNA sequence reads that are mapped to a transcript. How many RNA sequence read or fragments are mapped to your transcript. So then you will understand that for that particular transcript, what is the expression level or how it is abundant per kilobase of transcript length and is normalized to 1 million mapped reads in the sample. So this is your FPKM. RPKM is for million matrices is just for the single end RNA sequencing. So now you can come up. Sorry, there is another concept of a paired end sequencing and the single end sequencing. So what is the difference between those? So paired end sequencing is where you are starting the sequence from both the ends. Okay, from 5 prime as well as the 3 prime, and the single end is only. So there is some subtle difference in how the uh, library is prepared as well as how the analysis is being done for the paired end sequencing and the single end sequencing. So this is meant for the RPKM. Whenever you term the word RPKM, it is meant for the single end RNA sequencing, whereas FPKM is meant for the paired end sequencing. It counts up the total breads in a sample and then it divides the number by 1 million. Then per million scaling factor, so it normalizes your sequencing depth, giving you reads per billion and dividing the RPM values by the length of the gene in the kilobases. Then another which is being used very much extensively nowadays and it's being used in replace or substituting FPKM is your TPM, that is your transcripts per billion. So this is the expression unit for gene or transcript quantification in the RNA sequence data, and it normalizes the read count for transcript length and total map reads while accounting for the presence of alternative isoforms and your multiple reads. So TPM is now used. So if you check in the paper that TPM, so you can understand that it is the transcripts per million and is being used now uh, in place of FPKM. Okay, so the only difference, just for a note, that the difference is that it takes into account FPKM takes into account that it can be mapped to one fragment, so that since it is for the PNN sequencing, so it takes into account that so there is no duplication risks or there is no inappropriateness regarding that, and the difference is not and nothing else and between FPGA and RPM, and then it's the TPM. Yeah. What is it? So it is generally used in terms of for the PRDN sequencing for replacing FPKM, but I'll still cross-check that if, for single layer sequencing it can be used as well. Okay. Yeah, so these are the units like you measure in the abundance, or this is the measure of the how the transcript reads abundances or the what are the how many transcript reads are there for particular transcript. So these are the quantifier unit actually normalized RNA expression unit. Okay. Yeah. Yeah. So the your whenever what is that alternative algorithm with the for the particular sequence if there are some alternative forms are also present, okay, for that particular, especially in the when the transcribe put into it, so different forms for the same transcript leads to your same kind of transcript and same kind of expression. So that those alternative isoforms are also taken into account, which maps to the particular same transcript but different forms in the different forms. Yeah, so this is your kind of uh, homework or thing to thing. So each one is different paired PKM and TPM, yeah, for paired and RNA sequence. So this is the formula RPKM equal to number of reads which are mapped to your gene sequence, gene length, length of the gene sequence, and the total number of mapped reads to a sample. So total number of mapped reads means how many reads you could identify for that particular sample. So also you have to understand that there is the splicing effect and all those reads in every tissue or every sample you will not get if you are using a human or eukaryotic thing. So this is the total number of mapped reads to a sample, which is scaled by 1 million. Uh, Okay, so this is the next. So this is the number of reads. So this is the formula which I have mentioned. So just as a case of homework, you can consider, try to solve this. That consider a different the gene with the following information. Total number of mapped reads is this much. Length of the gene transcript in base pairs is this much. So you can understand that the base pairs are used for the understanding the DNA and the RNA sequences, base pairs or KBs, and the number of reads that map to this gene is 8,000. So, okay, so you can try to solve this thing on your own with this formula. So, what would be the RPKM help? Okay, so any questions in law? No, right? Okay. So now if we look at the different public repositories, so there have been quite a words of public repositories that have come up, which to help the public sharing, accessing the data, and helping the data. So this is, for example, one is your Genomic Data Commons, which is being funded by NCI. There is Sequence Read Archive. This is a common source. Uh, this is a common source of those, what you say, that repository where the sequences are deposited. So this is one. So let us look. So this is Genomic Data Commons, which is being comprehensive data repository that houses all your genomic, clinical, and imaging data from cancer research, which fosters your collaboration. Then there's your CPTAC. So the CPTAC is one tumor. So after the proteogenomics approaches came, I guess Sayan has one question. Here's this hand. Please. Yeah. Okay. 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 So here you can see that the RPKM's formula if you check. Yeah, kindly mute then you can ask after I yeah. So you can see that the RPKM is that in the number of reads. So you are trying to maintain for a particular gene sequence. So in the we are talking at the RNA level. So I think you all know the definition of a gene. So gene is that part of the in the eukaryotic you know that there's exon intron. So from the exonic part of that particular gene which provides you a functional RNA or functional protein. So the number of those reads which you are mapped to a particular gene sequence, so whatever gene sequence you are, whatever gene for the XYZ gene you are trying to find out what is its expression level. So how we do see that the how the number of reads are mapped to that particular gene sequence and the length of the gene sequence. So that is 500 base pairs. Then how much is there, or the length of the gene transcript? Then how much has been total number of mapped reads for the sample? So because you will that will lead to that you will count it every time, right? So here, for example, in this question, you can see that there are 2,000 length, but say every time you will count that. So that's why you are taking into the consideration of the gene length as well to normalize the thing, and then you are getting the total number of the mapped reads of a sample, that in that particular sample, other than this gene, how many are being mapped to the transcripts of the particular genes. So that is your reads per kilobase per million mapped reads. Okay, so reads, so you can pronounce it like that, in the one of the reads for that particular gene per kilo base. Okay, per kilo base, that is why the 1000 comes, that per kilo base of the length of the gene transcript per million mapped reads, per million mapped reads of the different genes that you have identified to the sample. So then it totally normalizes all the consideration of other gene sequences, other gene lengths, everything. I could make you understand, right? Yeah, so FPKM is takes into account the paired end sequence. So it is generally the twice of the RPKM that takes into both the transcript length and the total number of map reads in the sample. So it's generally being used for the paired end sequencing. Okay. Uh, yeah. Total number of map reads and the gene length. So how many reads, just read in this way, how many reads are mapped to a particular gene transcript, and what is the total length of the gene transcript, and how many total number of reads are mapped to all the gene transcripts. That is the thing. Yeah, so you just go through it once again, you will get understand. So it is a little bit complex, but if you go through it once again, you can understand it, I feel. Right? Yeah. Yeah, so this is one of the very comprehensive and very popular and emerging consortium of the. Yeah, so Ari has asked that he wants to study of fungus both in solid and with the most suitable method, Illumina, PASBA, and nanomer. Mass spectrometry is for the proteomic sequences. If you want to identify the protein sequences, then mass spectrometry. Otherwise, genomic sequences, you can go for Illumina, nanopore, anything. Yeah. I am not sure how that the genome of a fungus found in salt marsh would have any effect on that. But you can still go on in your Illumina Plasma and more things, and can see the pros and cons, and can decide on your that how much coverage you want, how much. In fact, you can try iron torrent as well. So every is has limitations on the coverage and the number of sequences and the number of samples. On the basis of that, you can decide on. So I have not included those for you to go and refer and study. Yeah. Okay, so there, so this CPTAG and has been very much useful and popular tool, which have aimed to improve the understanding of the cancer by integrating the large scale your proteome and the genome analysis, or whatever the genome analysis of the TCGA. So first, by the NCI, first thing after the whole genome sequencing and all, so there was a huge craze, and the TCGA launched and this to find out all the genomic sequences, but you can understand that it did not answer all the questions, and then came the consortium of the Clinical Proteomic Tumor Analysis Consortium, which is aiming to advance the cancer research by integrating the large scale proteome and genome analysis. Then another is the network which is known as the Apollo network. So you all can go and search that it is the collaboration among the NCI, National Cancer Institute, the Department of Defense, and the Department of Veterans Affairs. So these three are the have collaborated to integrate the proteogenomics into patient care, which helps to foster advancements into personalized medicine and the patient care. Now, as I mentioned, that it helps you to understand the personalized level in a personalized way that how things can be achieved. So each Apollo network is trying to make it existent into the patient care, but still there are many hindrances and there are still many gaps which the field needs to uh, comply complete. So this is one example of the recent publication. I would recommend or suggest you all to go to it to understand that what things can be done and how it can. So this is just their graphical abstract of this proteogenomic and metabolomic characterization of human glioblastoma from the CPTAC consortium. So in the as you know that the glioblastoma is one of the most aggressive forms of cancer, and to know its subtypes and the signatures, what is creating, how it is the differences are creating. So the EMT signatures, histone acetylation, immune subtypes, the metabolome. So these therapeutic options. So all these things could be captured between by using this approach. So if you go through it, as I have also then given in the colorectal cancer, so you all can go and identify, understand the concepts more clearly. So now after this, so I think all those heavy concepts. So I'll just do just a glimpse of the basic concepts from the bioinformatics. So I don't actually, I'm not aware of the level of the audience, so how much they are acquainted with. But if you all want some basic bioinformatics session or series as well, so you all can mention that as well, so that we can also make that basic bioinformatics thing, but I will just give a glimpse of it. So how, so actually that, as I mentioned, that from where the sequencing technologies or those things have evolved from the basic things. So similarly is the case with the data analysis. So if you want to understand the data analysis things, so you need to understand the basics of those things, of those repositories, how it was involved, and then only finally you can understand that, as I mentioned, that identification of SNPs and the variants, that but it all generated from here. So for example, in GenBank, so I'm not going and searching it, but you all can go and then retrieve the FASTA format. So FASTA is one kind of thing. So I'll just mention here that FASTA is both an algorithm to align the sequences, and also FASTA is a method to, is a format which starts with a greater than sign and which contains the sequences, be it amino acid sequences or the be it genomic sequences. Then if you then there are various tools. So for example, I think you all know NCBI, EMBL, and EDVJ, the DNA Data Bank of Japan, European Molecular Biology Laboratory, and the NCBI, National Center for Biotechnology Information. So, but you need to understand there are various tools, some run by EMBL and some by NCBI, but all those things needs to understand the sequences. So that's why comes the place that you need to have a conversion of the emboss. So emboss secret tool. So those who are familiar with it is fine. Those who know the basic bioinformatics, but those who uh, don't know, so you all can go and in the emboss secret try to uh, search that from FASTA format to NCBI gene format, or in this way you can play those things, and then you can identify and understand the things. So okay, so now just basic concept refresher that I think you all have many questions, but you can just I'd like to clear that there are some concepts or the terms from where only these complex concepts stand. From this basic foundation, only these complex concepts of SNPs, polymorphisms, quantifying expression, everything stands upon. So this is sequence. There are two important terms which we generally forget, is that the sequence homology and the sequence similarity. So, for example, there was I think many of you would be interested in the microbiome or those organism things. So they are, these terms are very much important, and don't want to just on the human thing. So the sequence homology comes from the term that the two sequences must have an evolutionary relationship, but the sequence similarity comes that may or may not have an evolutionary relationship. And the sequence homology also comes up from the part that can be, so in the sequence homology, there can be only one answer, yes, that it is the two sequences are either homologous or non-homologous. It cannot be a percentage in the form of the homologous. And then is the sequence similarity, there can be percentage, that this percent of the sequences are similar. So why I brought this thing here is that when we are saying of the SNPs and identification of those polymorphisms, how much the sequence is similar, the disease genome is how much similar with the normal genome. So if you think and link this thing, then the complex concepts would be also very much. What I mean is that it would be very much easy. It won't be so much difficult. So this is how the thing. So now these homologous for in the microbiome, or in this case, that the sequences can be either homologous, and that also depends on the particular zone of how much similarity sequences can be taken forward, and there are series of substitution matrices and all, which is beyond the scope for the discussion for today. And then is the another thing is for the your protein sequences. So sequence similarity, and there is another term called the sequence identity. So sequence similarity and identity same for DNA sequences, as we know that it is only ATGC, but in the protein sequences, what happens is that, so that's why you all asked that the synonymous mutation or the non-synonymous mutation. What is that? That the two protein sequences that have exact amino acid residues are called the sequence identity, but similarity means that the protein sequences have similar physicochemical properties. So whenever we study the complex things also, we have to understand that proteins can share similar physicochemical properties. So if the mutation is leading to the protein having similar physicochemical properties as well, if the amino acid has changed and the structure remains intact, then also the possibility of the having a detrimental effect is less. So the so identification of only mutations or only at the single amino acid variance level. Uh, so I wanted to stress out this fact that the identification at the single amino acid variance level is also not enough, because you have to understand that how things behave and function to finally identify that how it is affecting the change in your disease state. So then is your alignment. So I have told that the alignment and the reference to data analysis after your sequencing is done. So after your sequencing is done, you will align with the reference genome. So in the proteogenomics or in the genomics or the NGS sequence, what we are doing, we are taking the genome sequence and we are aligning to the reference genome. But these are used using various multiple complex tools. But how this concept of alignment came, which is important to know and understand uh, for better improved, or it might happen that you may come up with a better version of alignment of the tools that are present. So it is always necessary to understand that how we can align the tool. So first, there is two concepts of global alignment and the local alignment. So one is your alignment that happens over the entire range of sequence, the local alignment over local region of the sequence. So as I previously mentioned also, so this I am just giving up the revision of some basic concepts before we move on. And if you all want to have a series of those series where we have the complete data analysis as well as some basic competence, but you all need to mention in the groups. Then consider both the sequences. Global alignment considers the sequences to be of similar length in nature, but the local alignment can find your conserved sequences, and it does not take into account the length of the sequences. And the global alignment helps into best aligning the two closely related sequences, but the local alignment is for the divergent sequences. So if you having divergent sequences, then you can go for the local alignment. So this is of the uh, connection between the global alignment and the uh, local alignment. And then there are various methods, alignment algorithms. So these don't think if you all know these as well as that the basic concepts remain behind it same. So these are the some of those what. So first matrix dot matrix. Divergent means these are not closely related. These are the from the different groups or the different organisms, evolutionary distant organisms, to be precise. So the dot matrix method or the dynamic programming that determines global alignment or the Smith-Waterman local alignment, and the word method. So the whatever I think you all have in whatever level you are, so you have understood or heard of the term blast or have used it in your podcast in this. So this blast is nothing but it's a word method, or it's also known as a K-tuple method, which uses so the basic concept behind blast is that if the two sequences are similar, then there are certain number of words, the characters. So when after your sequence is generated, when we come to the analysis, we will start from the take those characters ATGC or your whatever sequence is there as a string and would try to match that what strings match with what. So the string two strings to be matched with it. So that from here the principles of the analysis, the concepts comes diverse. So that's ATGs is the two strings need to be matched with both the strings. So you need to have that. Okay, so at least some strings will be matching with that. So from there, k-tuple or mod method comes where it breaks into three fragments, and then the algorithm tries to map those three fragments with the entire length of the genome sequence. So this is a class which is generally used heavily, but for the uh, the DNA sequencing and the next generation sequencing and all those. I'll mention some of the tools and which we can discuss in further going on through this course. So this is a heuristic data mod method. So our biological databases help us to retrieve some information as well, and not only provides the data but provides the information. That is why it is also called as your functional databases or the derived databases. So this some of the heuristic database searching which using BLAST, the k-tuple method, BLAST and BLASTX, and coding Gs, and there are which we can discuss only if we have a some basic bioinformatics session, which we cannot discuss here in detail now. But those who are interested can go and search. But yes, obviously we can have. And so retrieving the whole exome or whole genome sequence. So there is one tool which is called SRA toolkit. So if you want to retrieve from this, as I mentioned, that there are different repositories, and like the sequence read archive is one of those repositories, open access repositories that contains various information about the genomic sequences that are happening all over the world and deposited here. So you can try to retrieve those. So it does not require any kind of thing. So, but it requires certain tools. So this is the toolkit. So I have just mentioned here SRA toolkit. So those who are enthusiastic or interested can try to retrieve any data sets and can try to analyze and can try to find out the sequences. So this is your SRA toolkit. Okay. So yeah. So this is your SRA toolkit is one such toolkit which will help you to download those data. So for those who are in a very basic level, I will suggest them to go into the BLAST and CBI gene, try to identify the different sequences in their genes and all those. And those who are in the advanced level can try with the SRA toolkit with the genome sequencing and can try with those methods. And fastq dump helps us to for the paired end sequencing. It will help us to uh, take out the two sequences, which is the command is used. So I think we can discuss it in other sessions. This is just for the concepts and the whatever things are there. And so for the proteogenomics approaches, so what are the how it is generally analyzed? If we from the analysis point of view, so we analyze it from the see, for example, they have taken from the genomic data commons, and then we try to align. If we don't have the personalized or the patient specific sequences, we try to aligned to HG38 human reference genome, which were previously have been updated with after HG19, and additional quality checks. So all the sequencing methods after analysis. So now you can understand that the sequencing approaches or the sequencing technologies are not so easy. So it always there's the issue of the reproducibility, the quality. So that's why always quality checks needs to be performed for first Q or the HTQ, first QC or HTQC, which helps us to understand the Q30 scores that okay, so the reads that have been called, the bases that have been called are not falsely called, or they are high probable that they are the right ones, that whatever the call has been made is correct or the right one. So these all quality checks needs to be performed, and then we need to analyze it into the whole genome, which gives us the BAM or binary alignment map. So what is the binary alignment map? Is that okay? The alignment has been done or not, but you cannot visualize those for integrated genome viewer or such tools, and then you can identify the copy number variants as I showed. The SNPs, CNVs, everything can be done using SNP if there are various tools, so which we can discuss in the subsequent things or the in different sessions altogether. Yeah, so how all these things were instigated. So I'll just mention these two things before ending. So the Human Genome Project has already done the completion of the Human Genome Project was helpful in understanding various things, but with the antibody-based proteomics and the mass spectrometry-based proteome draft of the human proteome, which we will discuss in the due course of this sessions, the mass spectrometry and how its role and how it helped it to understand the proteome of the human beings, or all the human proteome, and also it's getting advanced day by day. So that all instigated to integrate both these things and to get a better resolution at a small changes, understand those basic changes in the molecular level. So I'll request, recommend you all can go and study these papers, antibody-based proteomics and mass spectrometry-based draft of the human proteome, and can discuss that how you can understand. And then is the application, just briefing the application of the proteogenomics, so you can elucidate the complex interplay, and this helps to revolutionize the next theme of the personalized medicine. If you have heard of the terms personalized medicine and effective therapeutic targets, so this holds the potential to generate onto this. So this is a paper which is a recent use and views in the Nature, which says that the proteogenomic mapping sets stage for precision medicine. So you all can go and read through it. Yeah. So thank you for today's session, joining and attending today's session. So if you have any question or anything, yeah.